Okay, so I guess I'll talk about the one area of genomics that we haven't covered so far, which is DNA methylation, which is in many ways complementary to the gene expression type of profiling that we've heard about and the sort of histone modification analysis that Jason presented. And so, my lab's been working on this for many years. So what I thought I would do is just cover a few topics associated with DNA methylation, give some background, and cover some topics. And it's really more of an applied talk to try to address the question is why, how can we use DNA methylation to understand the systems that we're studying? So I'll just give some background, and then I'll talk about two applications. One is the use of DNA methylation for association studies, and the second is to use DNA methylation to understand the cell type, the components of mixtures, the cell type components of these mixtures. So DNA methylation, for those of you who are not so familiar with it, has to do with the addition of a methyl group to cytosines. And so on the space-filling image on the right, you can see what this does is it's a fairly small modification to DNA, but it's there are sort of some very stable techniques for sort of very precise techniques for studying the presence of these methyl marks, and so it's been studied for many decades. And as you can see, it doesn't have a big effect on the structure of DNA, but it has a subtle effect, and what we'll see is it has some. So one can think of it as sort of a mark on the DNA that tells you something about the state of the DNA and the state of the cell. So in order to have that mark, you need the enzymes that can place that mark on the DNA. And so there are two classes of enzymes that can place the methyl group onto the cytosines. The first are called de novo methyltransferases, and the second are called maintenance methyltransferases. So the distinction is that the de novo methyltransferases connect on completely unmethylated DNA as their substrate. So if you put in sort of naked DNA with no methylation and add these methyltransferases, they can start adding these methyl groups to the cytosines. The second group, on the other hand, is a system for maintaining the methylation. So once methylation has been placed during cell division, this mechanism can copy the methylation status of one strand onto the nascent strand. And so these two mechanisms ensure that, by and large, methylation is preserved through cell division, and sort of there's an inherent copying mechanism, and so that methylation is going to change slowly as cells divide and differentiate. So one of the questions we ask is, given that there are these DNA methyltransferases, how do they determine the patterns of DNA methylation that we see on the genomes? And so one way to address that question is to try to add these enzymes to organisms that don't have any DNA methylation. So basically, this is a case where you treat this as naked DNA, you add the methyltransferase, and you ask, where is the methylation placed? Of course, it's not naked DNA. So in this case, we look at Saccharomyces cerevisiae, and so Saccharomyces cerevisiae doesn't have endogenous DNA methylation, but it does have all the endogenous chromatin structure. So the chromatin of Saccharomyces cerevisiae is very similar to the chromatin of humans and mammals in general. And so we can ask whether some of the features within the chromatin impact the patterning of DNA methylation within this organism. And so to do that, what we did was we took one of these de novo methyltransferases. Obviously, it wouldn't make sense to put in the maintenance because there's nothing to maintain. So we need a de novo methyltransferase. And then we put it into yeast, overexpressed it, and then measured its impact by doing lots of different genomic type analyses. So we did this bisulfite sequencing, which, as I mentioned, is the primary, sort of the gold standard for measuring DNA methylation. And what bisulfite sequencing does is, rather than sequencing DNA in a traditional way, it sequences DNA in which we've used sodium bisulfite to convert cytosines to uracils. But this reaction is inhibited by the methylation of the cytosine. So it only measures that conversion when the cytosines are unmethylated. So if we measure these conversion events and we know what the reference sequence is, we can then infer what the methylation status of the genome is. But then we also asked, is the patterning of methylation affected by other things? So could it be affected by nucleosomes and DNA accessibility? So for that, we use micrococcal nuclease digested DNA sequencing to see where the nucleosomes are positioned. We also looked at RNA sequencing to see if there was some effect between transcription and the patterning of DNA methylation. And we also looked at chip sequencing to see if certain histone modifications might be tightly correlated with DNA methylation. And so when we do all that, we get images that look like this. So I guess there was a pointer here somewhere. But, top one. OK, yeah. So the top track here shows DNA methylation. So the first thing we see is that when we add these mammalian DNA methyltransferases into an organism that doesn't have DNA methylation, they are in fact able to methylate the DNA. And I'm not showing a lot of the background data, but basically these methyltransferases will only methylate CpG dinucleotides. And so you can see when these bars are high, it means the methylation is high. And when the bars are negative, it means the methylation is on the reverse strand. And then what we can see, we did chip on the de novo methyltransferase itself, and we can see, not surprisingly, there's a strong correlation between the binding of the methyltransferase and the DNA methylation. So where we see binding of this methyltransferase to the DNA, we see high levels of methylation. But then we see there's also some strong correlations with histone marks. So we see that this particular mark, H3K36 trimethylation, which typically marks transcriptionally active regions of the gene, is strongly correlated with DNA methylation, whereas this mark, H3K4, which typically marks the promoters of genes, seems to be anti-correlated with the DNA methylation. And then there doesn't seem to be any really significant interaction between the transcriptional state and the methylation. And so when we put this data all together, we get sort of a complex pattern of correlations, but the strongest correlations we see are that there are very negative correlations between the presence of this H3K4 trimethyl mark and DNA methylation, and that there are strong correlations between the presence of this K36 trimethyl mark and DNA methylation. And so what we asked was whether these two marks are indeed sufficient to determine the patterning of DNA methylation. And so for that, we built some simple regression models where we ask, can we model the DNA methylation signal in terms of the presence and the intensity of these two marks, H3K4 and H3K36. And we see that if we try to model it with the marks independently, it does OK. But if we combine the two marks, we get a very strong agreement between our model and the DNA methylation pattern. And then if we add additional data, all the other data we collected, the transcription, the nucleosome position, et cetera, we don't really improve the model. So what this tells us is that DNA methylation is by and large, or at least to first order, reading off the chromatin state of the cell. So whatever the particular state of K4 and K36 marks in the cell are then going to determine the pattern of DNA methylation in that particular cell. And so we asked, OK, this is what happens in yeast, but yeast is not a mammalian system. So what happens if we do the same experiment in a mammalian system? And so we collected data. The data collection was done in the Plath lab, where they looked at many different histone marks. In this particular example, these are embryonic fibroblasts. And we asked the same question. We made a simple logistic regression model where we asked, can you explain the methylation state of each cytosine as a linear combination of the abundance of reads associated with each of these different histone marks? And what we find is that, again, with just including the K4 and the K36, we get a very good agreement between the presence of DNA methylation and the presence of these states. And if you look at the actual tracks, it looks quite remarkable. This is the actual observed DNA methylation. In this case, it's in embryonic stem cells. And this is the predicted DNA methylation just using the K4 and K36 marks. And to an untrained eye, these two signals look essentially identical. So this is saying that by and large, even in mammalian cells, the DNA methylation is reading off the presence of these K4 and K36 marks in the cell. So then that begs the question is why would you want to look at DNA methylation as opposed to looking at the histone marks? What's the relative advantage of studying one over the other? And what I would argue is that the kinetics of the two processes are quite different. So one way we can do that is, again, using data from the Plath lab and using the Chrome HMM states that Jason presented, we can break down the chromatin states in these mouse embryonic fibroblasts, and then the change in these states after 24 hours when these embryonic fibroblasts are starting to reprogram towards an embryonic pluripotent state using the Yamanaka factors. So this is one of the primary areas of research in the Plath lab. And so what we find is that after 24 hours, so this shows the occupancy of states in the fibroblasts and the occupancy of states after 24 hours. And you can see that as fibroblasts undergo this differentiation after 24 hours, there are significant changes in chromatin. So this is depicted by the fact that we don't just have diagonals here. We have many examples where certain enhancer states are being reprogrammed to transcriptional states or promoters to enhancers, et cetera, et cetera. So there's quite a robust change in the chromatin of these cells after 24 hours. In contrast, when you look at the DNA methylation of the same cells, there's essentially no change whatsoever. So what this tells us is that although the DNA methylation is reading off the chromatin, it's doing it on a different time scale, right? So the chromatin states are changing quite rapidly by comparison, whereas the DNA methylation takes a long time to catch up. So what you can think of is that the DNA methylation is somehow a genomic readoff of a time-averaged histone modification state. And after many passages of cells, it eventually equilibrates and reads off that mark, but during the periods of transition, when those marks are changing, it won't accurately read off the marks. So this sort of brings me to this notion of genomic timescales, right? So when we think of genomics, we also have to think of time, because each of these marks is actually telling us things on different timescales, right? So you can imagine, we have timescales that are invariant, and then timescales of weeks, hours, and then timescales of minutes. And we all know that gene expression is extremely sensitive as a genomic readout on a very, very short time scale. So people who do measurements of cell induction in response to cytokines and other things, they know that you have to start looking after just a few minutes to see the very first transcriptional events in the cell. And as I showed you, some of these histone modifications, although they're not quite as fast as transcriptional, they still start to accumulate even after 24 hours. In contrast, we think the DNA methylation is a much, much slower process, so it's going to take days or weeks for it to equilibrate to a change. So if we do an example where we take cells, expose them to a cytokine, and look at 48 hours, we won't see absolutely any changes in DNA methylation, or no real robust changes. And obviously, at the other opposite end of the spectrum is the genetics, which is completely invariant, with the exception of a few mutations here and there. So we can think of as this sort of the opposite end of the spectrum from gene expression. So given all this, what kind of opportunities does DNA methylation profiling present? So we know that there's this sort of hierarchical structure of genomic data that we can collect to study our systems, and there's a great interest in studying the underlying genetics. And as we heard, there's a very enormous amounts of effort in studying the transcriptome, and the epigenome is sort of straddling these two systems, both in terms of timescales and in terms of dependencies. So we know that the epigenome depends on the genetics, and I'll show you some data, but that, it's fairly clear that the genetic differences within a population can drive some of the underlying epigenetic differences. And we know, as I showed you, there's some correlation between the epigenome and the transcriptome. But of course, the epigenome, unlike the genome, and the transcriptome is also going to respond to slowly varying environmental cues that are applied to the system. So one possible sweet spot for these epigenetic profiling is that it can tell us something about time average changes that are occurring in the environment and how these are impacting our system. And so in particular, one thing one can look at is diet, right? So we know that diet is something that can affect the physiology of an organism. And so together with the Lucis lab, we used DNA methylation to profile how the dietary response can be measured at the molecular level through DNA methylation. And so he has this system called the Hybrid Mouse Diversity Panel, which consists of 100 different mouse strains. They're all homozygous, so one can go back and do essentially an infinite number of experiments on strains with the same genetic backgrounds. And in this particular set of experiments, he fed these mice different diets and then measured the weight gain in these mice. And he found that some mice don't gain any weight when you feed them different diets, when you go from sort of a standard diet to a high-fat, high-calorie diet. But other mice become extremely obese. So on this end of the spectrum, these mice, their weight gain was about 600%. So these mice are really getting enormously fat. So there's clearly a strong difference in the response of these mice to the diets. And so this kind of experimental design can let us sort of dissect which of the responses to diet are due to the genetics and what the epigenetic consequence of these responses to diets are, because we can compare the dietary response between the chow and the high fat within mice of the same strains to tease out, what effects does the diet have on DNA methylation independently of genetics. So the first thing to note is that within the system, the genetics trumps the epigenetics. So if we do, in this case, we profiled 6 different strains. And again, these strains are fed different diets. So there's the chow diet in red and the high-fat diet in blue. And if you see if you cluster this data, so we typically measure on the order about several 100,000 CPG sites within these genomes. And if we do a simple clustering, we find that the clusters correspond to the different genetic backgrounds. So again, a lot of the differences in DNA methylation between strains are going to be driven by the differences in their underlying genetic background. But you can see that within certain strains, there's also sort of clustering by diet. And so there's clearly going to be some epigenetic response to the different diets across these different mice. Although, again, the signal is going to be smaller than the genetic effect. And so we can think of this as a way to study gene by environment interactions where we have differences that are due to strains, differences in DNA methylation across these mice that are due to strains, which are going to be the most abundant, and then others that are due to diet, and then some are going to be sort of interaction terms where the diet effect depends on the strain. So we'll see a diet effect in certain strains, but not in others. And this sort of shows the relative abundance of these types. And you can see that, again, the diet and interaction terms are significantly smaller than the strain effects. But interestingly, if we take these diet effects, which, again, we're sort of then taking out the genetics from the equation and just asking what is the effect of the diet on the DNA methylation, we find that it has a very strong enrichment for sites that are near genes associated with lipid homeostasis. So not surprisingly, there's some interplay there between the DNA methylation and the activation of genes. And as we said, because DNA methylation is patterned by sort of the state of the promoters and the transcriptional and the transcribed region of genes, it's not surprising that a lot of these changes are associated with genes that might be undergoing dramatic differences in expression as a result of their diet. And specifically, when we look at one gene that keeps coming up in our analyses is this fatty acid synthase gene. So the fatty acid synthase gene shows dramatic differences in methylation and gene expression in response to these diets. And what we know is that the fatty acid synthase gene is in sort of a precursor to the production of a lot of the storage lipids. So when we store fats in our adipose cells, a lot of that storage is occurring in the form of triacylglycerols and other types of lipids, and the fatty acid synthase gene is sort of upstream of that and enabling that kind of lipid storage. So it's a critical component of our ability to store fats, a modulator of our lipid storage in these cells. And so the next question we asked is, well, okay, so it's not surprising that the mice are responding at the epigenetic level in regions associated with these lipid genes, and in particular the fatty acid synthase gene, but what's happening in more complex systems like human populations? And so to study that, we looked at this population of 10,000 individuals from Kuopio, Finland, and it's called the metabolic syndrome in men population. And the difficulty of the reason we start with mice and then go to humans is that in the humans we can't really control either the environment or the genetics. So we have this large population where both the environment, people's dietary habits are very different, and their genetics are going to be different within the population. And so we can't really separate out these two effects. And so what we can do, though, is try to see if any of the effects we see in the mouse are reproduced in the human. And in this particular population, we have detailed phenotypic information about their sort of lipid abundance, glucose, insulin, and these kind of measurements. But we also have environmental information about their diets and smoking and drinking habits. And in particular, for this study, we looked at the adipose tissue that was collected from these individuals, and again performed DNA methylation profiling in the same way that we did for the mice. And so what we do here is what we call epigenome-wide association studies. So basically, it's the same as the traditional association studies done with genetics, but instead of here having the underlying polymorphisms, the different alleles in the population, here we have the methylation levels that are observed across our population. So we can think of methylation at any one site as varying between 0 and 1, so completely unmethylated or fully methylated. And then we have these traits that are measured, and then we look for cases where there's a strong association between the traits and the methylation level, and then we can report these as significant p-values. And again, all the infrastructure for doing these type of calculations is the same as in the GWAS analysis. And so when we did this study, we found these are the different traits that we looked at, and each dot here represents a significant association between a location in a CPG in the genome and that particular trait. And what we found is this study had only a couple hundred individuals, so we found about 70 very significant associations, but these were clustered in a smaller number of loci. And in particular, we found 3 regions in the genome that have very strong associations with multiple traits. And one of these, and the strongest of all, again, was the fatty acid synthase gene. And when we look at this particular gene, we see that there's a very strong association. So these are the p-values, 10 to the minus 10, between the methylation of the fatty acid synthase gene and the BMI of the individual, their waist circumference, their weight, and plasma insulin levels, as well as other phenotypes that we can measure. So somehow this gene, both in the mice and the human, seems to be sort of a molecular readout of the physiological state of the individual, and potentially a readout of their diet and the amount of lipids that they've accumulated as a result of that diet. And when we actually look at one single site, there are many sites in the gene that are associated with the phenotypes, but here's just one. And we see that, so this is again the same kind of plot where we see the methylation on the x-axis, and in this case the weight on the y-axis. And again, these parameters have all been transformed, so this is not the actual weight in any normal measure. But you can see that there's a strong correlation. The R squared is 0.12, so this suggests that the methylation of the single site is capturing about 12% of the variance in the weight of our population. And so this, again, tells us that with just a limited number of markers, we could actually infer what the weight of that individual is, so that the environmental effects on these sites are quite strong, and their ability to then read off what the environmental state is, what the physiological state is, are quite accurate. So in the last part of the talk, I just wanted to transition to this last question. When we look at DNA methylation changes in these kind of studies, they could be caused by two effects, right? One effect is what we discussed, is potentially changes in the transcription and the chromatin of these different individuals. But another effect that can impact DNA methylation is sample heterogeneity. So we know that within samples like adipose biopsies, such as the one we're collecting, we don't have pure adipocytes, right? We're not doing any kind of cell sorting or anything like that. So there's going to be a mixture of different cell types. There might be endothelial cells, there might be hematopoietic cells, there might be other sort of cells that are within that biopsy. And so because each cells might have distinct methylation patterns, it's possible that these different abundance of different cell types could impact the methylation patterns we're seeing. And so to try to address that question, we tried to build a system for identifying what type of cells are actually present within these biopsies. And so we started off looking at the hematopoietic cells because, well, we had good reference data for those, and because you know, the differences, their methylation differences have been well characterized. So overall, we know that as sort of hematopoietic stem cells differentiate into the different lineages, there are distinct sites within these cells that have specific methylation changes. And so by looking at the methylation profiles of the fully differentiated cells, we can find sites that are specific to that one type of, that one lineage. And so having that information, then we can build a system for inferring what the abundance of these different types of cells is within a biopsy. So the idea is quite simple. You start with reference methylation data. So we have to collect methylation data from cells, single types of cells that are sorted, that are likely to be present within our mixture. And then once we have these cells, we want to identify the sites within these cells, within these methylomes that are specific to each one of the types of cells. And then we want to input the heterogeneous methylation data that comes from our biopsies, such as the adipose. And then we perform linear regression, in this case non-negative linear regression, to try to infer what the relative contribution of each of these individual cell types is to the mixed data that we collect. And so at the end, we can then estimate the proportion of each of these cell types within our mixture. So this just shows a map. Here are six different cell types that are quite abundant within the blood. And so we collected reference methylomes for each of these, and then we tried to identify sites that were specifically demethylated in one of these cell types compared to all the others. So blue here indicates that the site has low methylation, and red symposium will go on. So this, for instance, shows that within B cells, there are a set of several dozen sites that are specifically demethylated in B cells, but they're hypermethylated in all the other cells within the hematopoietic mixture. And so similarly for all other cell types. And so using these specific sites, then we can try to deconvolute mixtures of cells. And so one way we did this is by creating synthetic mixtures where we know the answer. And so Dennis Montoya in my lab generated 6 different mixtures of these 6 different cell types. And some were overrepresenting T cells, some were overrepresenting B cells, etc. And then we did the DNA methylation profiling on these mixtures with known concentrations, and we tried to infer whether we could then predict what those relative abundance of each cell type in the mixture was. And the approach works remarkably well over quite a broad range. So this shows the correlation between the actual percentage that we know went into the mixture and the inferred percentage that we obtained using this system. And you can see that the two are very, very strongly correlated, and for all 6 cell types over quite a broad range, even down to just a few percent, up to mixtures that contain nearly 100% of that particular cell type. So what we find is that DNA methylation is far more accurate, in fact, than using gene expression for doing this type of deconvolution, in part because you can identify these cell type specific sites, and in part because, unlike gene expression where the signal comes from the change in abundance, here the signal just comes from the change in methylation, so each cell is only contributing the same amount to the signal. And so it's sort of an inherently simpler problem. And so just as a final application, we went down the hall and we asked our colleague Don Cohn to give us some samples of blood from people with severe compromised immune deficiency. And we know that in these individuals, when they're born, they have deficiencies in their immune system, so they typically lack B cells or T cells. And in the Cohn lab, they use gene therapy to try to reconstitute that immune system. And so he had access to blood from patients who had undergone this immune cell reconstitution, and he knew which ones had had sort of more successful reconstitution of their immune system and which ones had had a less successful. And so we took the blood and we profiled it, and we found that a couple of the patients looked quite normal, and in fact these are the ones that he would consider to have been successfully reconstituted, where the gene therapy has really brought them back to a near normal state of immune state, whereas these samples clearly were still deficient in CD8 and B cells, so they had deficiencies in their lymphocyte abundance, and it was clearly evident from this deconvolution. And these were the ones that he knows, had sort of an imperfect reconstitution of their immune system, and they still have to undergo other types of therapies to manage that defect. So, even in real cases, this approach can accurately quantify sort of deviations of immune cell abundances from what you would expect in a normal individual. So just to summarize what I went through today, so the first thing I didn't tell you, I told you a lot about the patterning and the uses of DNA methylation, but I didn't tell you anything about what the function of DNA methylation is in the cell. And that's because, surprisingly, that's sort of one of the least understood features of this whole thing. We can very accurately measure DNA methylation, and we've found many ways to use DNA methylation, as I illustrated, but we still don't really have a very good grasp on the function of DNA methylation. There are some genes, some proteins that specifically bind methylated sites and others that specifically bind unmethylated sites, but these tend to have very subtle functions, and so labs that are trying to study the functional effects of this do not get very clear. So in some sense, DNA methylation is there for genomicists to use as a biomarker rather than for biologists to understand what its function is. So even though we still don't have a good grasp on the function, we have a very good grasp on how it's patterned, how it's how it interacts with chromatin, and that we have some sense of the kinetics, that these patterns are on a much sort of change on a much slower scale than both gene expression or histone modifications, and that these slow changes can then read off again slow environmental changes such as diet and the physiological state of an organism. and that these environmental effects can then be read off on a very few, sort of a limited number of sites in the genome that show a very strong response to these environmental effects. And in particular, I showed you that fatty acid synthase, which is one of the main genes for storing fats within our adipocytes, is very responsive. Its methylation level is telling us a lot about the human weight and metabolic state. And then finally, I told you a little bit about the fact that also cell type heterogeneity can contribute to differences in DNA methylation, and so we can leverage that aspect as well to accurately deconvolve mixtures of cells to identify what the individual cell types within the mixture are and what their relative abundance are. So in the future, we're sort of progressing along these lines. So we're focusing on using this sort of power of studying individual cell types, and also within this sort of systems approach of looking at large physiological responses, so that then we can try to explain some of these significant physiological responses in terms of modulation of individual cell types. So it can bring us down all the way from the environment down to individual cell types and how they may be modulating these effects. And so some of the people in my lab who have done this work are listed here. And then this is a collaboration that we've been going on with the Jacobson and Lucis lab, and also with the lab in Finland that's generated this MetSim cohort and sent us the samples. So with that, I'll stop.